So here 10-year-old child presents with severe sunburn, that is your xeroderma pigmentosum, which is a nucleotide excision repair pathway defect. Now let's take a look at the next question. 3-year-old child, developmental delay, 4 base pair, that is functional enzyme. So here this question is actually a bit tough because this question deals with a frame shift mutation. So now we are coming to genetics proper. So welcome back. 3-year-old child, developmental delay, failure to drive, GAA gene, completely non-functional. So we'll look at 2 questions together. Next question is, you have a 5-year-old child, progressive developmental delay, mutation in ARSA gene. So now we are dealing with genetics, okay? So in genetics, before we begin, there are a few terms that I want you to know. Now, when you're talking about genetic disorder, there can either be something called a Mendelian disorder. That is mutation in one gene cause a massive effect. That is your Mendelian disorder. Then you have your chromosomal disorder. Then you have your complex multigenic disorder. That is about genetics. So, what is the meaning of the word penetrance or penetrance? Penetrant. Okay. So, what is penetrant? So, if a mutation is present, if the disease happens, that is, if mutation is present and the disease happens, that mutation is said to have a high penetrance. So, a single mutation resulting in a single disease, whatever the disease may be, that is called penetration. The ability of a mutation to cause a disorder is called penetrance. Now, that is penetrance. So, what is mutation? Mutation is a sudden, inheritable or acquired permanent change in DNA. Now, mutations can happen in several sequences, like several areas. So, first you have a point mutation that is happening within the coding sequences. Okay. So, a point mutation is something that is, point mutation means only a single nucleotide is getting changed and that change is occurring in a coding sequence. Now, that point mutation can be 2 types. It can either be a missense mutation or it can be a nonsense mutation. Okay. So, this we'll deal with nonsense mutation first. Suppose whatever GAGG, whatever it may be, a mutation happens, and as a result of a mutation, if a stop codon comes, that is the DNA transcription the DNA translation stops happening at that point. That is, instead of a normal nucleotide, or a normal protein that is being produced, instead of that, what is happening? You have a stop codon. So that is called a nonsense mutation. Now, you can have nonsense mutation and you can have missense mutation as well. Now, missense mutation can be of 2 types. It can either be a conservative mutation or a non-conservative mutation. So, missense mutation is in the codon, one of the nucleotide gets changed. Now, if the nucleotide, or because of the mutation, if the nucleotide that is formed is the same, that is called a conservative mutation. So even though a mutation has taken place, if the same nucleotide is getting formed, that is a conservative mutation. Instead, if because of a mutation, a different nucleotide is getting formed, that is a non-conservative mutation. For example, you have your sickle cell anemia. Now, in sickle cell anemia, what do you have? You have the glutamic acid being replaced by valine. So that causes a difference in the change, and that is sickle cell anemia. Now, that is mutation within the coding sequence. Now, suppose the mutation happens within non-coding sequence. Mutation happens within non-coding sequence. So here you can have the non-coding sequence can be a promoter area or some other areas. And if the mutation happens there, it can also result in disease. For example, thalassemia. Now, if the beta globin chains that is getting produced, if there is a mutation in the promoter region and all, that causes thalassemia. Now, your third type is your deletions and insertion. Okay, so deletion and insertion is, what do you mean? So here you have some GAA, GAC, GA, GUA, GTA or something like that. So if nucleotides are either added or subtracted in groups of 3. So here, suppose this is your codon. If here, if it is GAA that is there, that is in group of 3, a mutation is happening. The reading frame of that mutation is not going to change, because how does reading happen? In 3 codon, then 3 codon, then 3 codon, that is in the codon, then 3 nucleotide, 3 nucleotide, 3 nucleotide and 3 nucleotide. If group of 3 or multiple of 3 is happening, the reading frame does not shift. But if instead of 3, if only 1 or 2 nucleotides are changed, that is, if here instead, if you can have an A here or a T here, so this reading frame is always in this 3, 3, 3, 3 like that, no? So here it is okay, here it is okay. So here in GTA, if one A is added or one T is added, the entire frame is going to shift. That is called your frame shift mutation. And an example of a frame shift mutation is your Pompey's disease. Okay. So then again, you can have some alterations in trinucleotide repeats. A very common example is your FMR1 gene, that is your familial mental retardation 1 gene. Okay. Then 2 other words I want you to know is, first one is pleiotropism. So what is pleiotropism? Pleiotropism essentially means that a single mutant gene leads to many side effects. That is pleiotropism. So what is genetic heterogeneity? Genetic heterogeneity means that mutation at several areas lead to a single disease. Okay. So now let's take a look at the question. 4 base pair deletion in GAAG. I said earlier it is Pompey's disease. So what I want you to know is, this hepatomegaly, failure to thrive, developmental delay, it can be in any of these. Any of these 4 options can present as this. But the fact that it is a 4 base pair deletion and it is a frame shift deletion, that says the answer is Pompey's disease. Okay. So next one, 5-year-old child, progressive developmental delay, ataxia, seizures. Again, these things can happen in any of these. The question will tell you the answer. That is, if lipid-laden macrophages, foamy cells, ARSA gene, that is your MLD. Okay.